Dear children and the parents, I'm going to start the new program which is called Homework with Online Guide HOG program. So I will take 1 minute to introduce the program. In this program, every week, a student get a small homework like this, just only the 2 pages worksheet, and it will not take more than 1 hour or maximum 1 hour 30 minutes, 1.5 hour time. So how it works? The homework is given in the classroom, and then student should take this home. They have 1 week to complete this. During completion of this work, they have complete online video like this, describing the question. So you can read the question. If you can answer the question, no problem, you can answer. But if you feel it's difficult to answer, you don't want to bother about parents or you don't want to bother about any people, you just stop. You see the pause sign is coming with this video, and you stop and you try to do it. If you can't do this one, continue the video and watch the solution, and then you can complete. So after complete this homework, you have to hand over to me in the next class. Then I will collect the homework. I will check this again, and I will give a feedback, quick feedback on the homework sheet, and then I return the homework to you. This is going to happen in every week. So it's a small work, but it will make a big difference at the end. Right? So let's start the first HOG program, and this is the question we're going to learn today. So ready? Okay. So we'll start. Right, let's start the question. The first question is, apples contain an enzyme called phenol oxidase. When apple tissue is exposed to oxygen in the air, this enzyme turns the apple tissue brown color. Right, this is a normal phenomenon. We have many times observed this kind of a thing. Some fruits and vegetables, they have this phenol oxidase enzyme. After we cut the fruits or vegetable, we can't keep longer. They get brownish color. This is due to enzyme activity. So then this question is based on enzyme. Okay, next one. A student uses this method to investigate the effect of pH on the time taken for apple tissue to turn brown. So then the main concern is effect of pH and how long will it take the apples to to brown when the pH change. So let's see how the pH change. The student method, now mash is this. Mash an apple to produce a pulp. So simply blend the apple tissues to make the pulp and place 20 gram of pulp inside this beaker. And next thing is, you don't want to worry about this word buffer. Okay. Start. So this word buffer is used here. So at the moment, don't worry too much about this word and just leave it, and we just concern about the pH 7. So then the student maintain at pH 7. He used some pH 7 solution, then we call pH 7 buffer, to make the solution 7, pH 7. And next one, check the color of the pulp every 5 minutes. Then after adding the pulp and the pH buffer, then the student check color at every 5 minutes. Record the time when the pulp turns brown. So then the student record the time when it becomes brown color. And student repeat this experiment for different pH, 3, 5, 9 and 11. pH 3, 5, 9 and pH 11. Did you understand the method? Simply take the apple pulp and the student add solution to make different pH, 3, 5, 7, 9 and 11. And student check the color, time taken to color change from normal fresh apple color to brown color. Okay, these are the student result. So you can see this kind of a graph is called bar graph. Look at the y-axis. You can see in y-axis, it's a time taken for apple pulp to turn brown in minutes. So just he measured every 5 minutes, and then he recorded the result. Now the result convert into a graph. So you can see pH 3 it took long time, and pH 5 again quite long, and pH 7 it's very short, pH 9 a little bit longer, and pH 11 it's again longer, right? Now calculate the percentage increase in time taken for the apple pulp to turn brown at pH 11 compared with the time taken at pH 7. So if you think you can manage this question, just pause the video and calculate it, and then check whether it works or not. If not, you can correct it, right? So, okay, so then I will keep on continuing explaining how you can calculate the percentage increase. Right now, I take the graph again. So when I take the graph, I would like to show you the two pH that we're going to calculate the difference, right? pH 7, and we want to read the exact value on the y-axis, and pH 11, we want to record exact value on y-axis. What you have to do, you take a ruler and you draw a line from pH 7 top of the bar to the y-axis. As you can see in this picture, take the ruler and take a pencil and keep the ruler on the top of the bar of the pH 7. And then you can draw a line to y-axis. Same thing you can do. You can take a ruler again and keep on the top of the bar, and then draw a line like this using a ruler and the pencil. Now you see, yeah, that's right. So now you can see the pH 7, it's just going on the y-axis, but you can't see any values to quickly take. But if you take on the pH 11, it's just going exactly on 75. So that means you take this reading as 75 minutes. Fine. So now we're going to learn how to take the value from y-axis if we don't see exact values, right? So look carefully how I'm going to do that. Now, this is called y-axis, and it is called linear. Why I call linear? Values are spaced equally. What does it mean? 0 to 25 is different is 25. 25 to 50, different is 25. So any value in this y-axis, the difference is 25. This is called linear. So if the values are spaced equally, we call it linear. So it is very important even in the future when you're going to draw graph. So you should use linear scale, otherwise you don't get marked. So now you learn one thing, what is the meaning of linear scale in a graph. Then the second thing, you're going to look the 0 to 25. I just make it bigger now. 0 to 25, the square, it's a big square, so it has 25 values, right? Or other words, 25 minutes. Then we also have a small square like this, and the value of the small square is not given in the graph. So we have to calculate. Now what we do, we just going to count how many small squares within this 25 minutes or 25 values. So how many small squares we can find within this 25? So how you do that? 25 divide by 5, because you can see 5 small squares make 25. So then 25 divided by 5 small squares, you get 5, right? You get 5. That means small square value is 5. So now we learn another thing, how to read the values on y-axis. Okay, now we can read it. Again, you do draw a pencil line. Now we know it is 5. We don't see, but now we know small square is 5. If it is going a 2 small square, it is 10. If it is going 3 small square, it is 15. So like that, we can calculate. Now that one is 75. Now, next thing, look very carefully. So I got this value 5 and 75. After that, so I'm going to show you something to remember. That means you need to really remember this formula. So this is one of the best formula always used in maths and biology and maybe chemistry and physics also. So you need to remember this, right? So now we know the difference. 75 to 5 is 70. 75 minutes, the first reading, and 5 is the second reading. 75 minus 5, we get 70. Right now, this is the formula. Percentage change formula, percentage increase or percentage decrease, we take this formula. Final value minus initial value, simply the difference that you can see above. 75 minus 5 is 70, right? That's a final value minus initial value. Divide by initial value into 100. So now I write it 70 divided by initial value is 5 into 100. So you get 1400%. Now the answer is 1400%, right? 1400% is the answer, percentage change. So you will get more similar calculation in the future. Use this formula, final value minus initial value divided by initial value into 100. Remember the formula. So next time, whenever you get math, you will do it, definitely, right? Okay, let's continue to the next level. Explain why the change in pH affects the time taken for the apple pulp to turn brown. So now we see different pH, we have a different time. So pH 7, it's very fast. And pH 3, it's very slow. And pH 11 also very slow. Now we are coming slowly to the questions. How and why? That means when we have a pH 7, it's faster. That means enzyme work faster. But pH increase or decrease, enzyme gets slower, right? So I will quickly revise you what is the theory behind this. Okay, so let's check in this case, the enzyme. You can see here, the enzyme has a specific shape on its top here, or enzyme has a special site, which is special shape, and you can see here that is called active site. Onto the active site, you can see now substrates bind. Here you can see 2 substrates and one active site, but active site has 2 specific phase and the substrate bind. Now, after substrate bind to the active site, what will happen? Substrate convert into products. Then products leave from the enzyme. Enzyme is again available to catalyze or speed up next reaction. Right, now we have learned how enzyme work Number one, enzyme has active site. Number two, substrate bind to the active site. Then number three, substrate converts into products. Number four, enzyme is again available. We also learn what are the factors that can change enzyme's activity. There are two factors. Number one, temperature. So temperature can change enzyme activity. Number two, pH. pH can also increase or decrease enzyme activity. So here you can see the graph, the pH 1 to 14, the rate of reaction increase, and the highest level is called optimum pH, then again decrease. So why this happen? Low pH and high pH, the proteins destroyed or shape change, active site change shape. This is called denaturation or denatured proteins, enzymes denatured, denaturation. That means high pH and the low pH, the two ends, when you go to two ends, the proteins structure destroyed. That means enzyme active site change. This is called denature. And the second one is the temperature. The graph looks similar but little bit different. It's not that symmetrical. And when it's come to cold, it is a bit more. So when it is cold, enzyme activity is less because kinetic energy is less. They move not much faster. Then again, the highest rate is called optimum temperature. Then come to higher temperature, the heat can again destroy the proteins or denature the enzyme active site. So that's very important two things to revise. Now, if you look at this graph, the optimum pH is 7 because rate of reaction is the highest. Enzyme activity is the highest. It quickly changes color because enzyme quickly change color. So then pH is the optimum. But if you go to the low pH, so time taken, more time taken, and higher pH, more time taken. So the model answer for this is the optimum pH for enzyme activity is 7. Change of the pH can lead to the denaturation of the enzyme, resulting in the change of the active site. Substrate cannot bind to the active site. So enzyme activity reduced. So I highlighted the main point: optimum pH, active site, denaturation, and the substrate cannot bind. So I'm also sharing with you the marking scheme point, how we can write a model answer to match to the marking scheme point. So optimum pH is 7. Second one, pH causes enzyme to denature, shape of enzyme or active site change, substrate does not fit into the active site. Now we look the next part of the question, C part. Explain how the student could modify this method to give a more accurate measure of time taken for the apple pulp to turn brown. So now we want to propose a method that we can measure accurately the time taken to change the color into brown. Okay, so now you have to think like this. So look at the apple on top. That's a fresh. And then look at apple on the bottom. So that start to change the color and the shapes like this. So when we measure something from our eye, from human eye, especially color like things, that is subjective. That means I might see that it is a end point, but someone will say, no, that is not brown, I will wait more. Another person will say, okay, I will wait more. So now everybody has to judge the end point, that is a brown color. So this kind of measurements are subjective because when you look color like things, it's person to person, maybe slightly different the color. So then if we want to make accurately, we would like to propose some tech or some devices or some kind of a technology or some kind of a modern method, not just human eye, or maybe at least combination of a human eye with something. So in that case, one of the basic thing, not high tech, the basic thing, we are trying to match with the color chart. That means we have several brown colors, shades of brown colors, and we mark one brown color. Okay, these shades, if I see and I match with the apple, then I stop. That's called match with the color chart. So there are other methods. We leave these things. We can learn in the future some of the method, but at the beginning, so we can rely on the color chart. So then you can match with the color chart. That means, so when you see the apple and you check the color chart, whether it is come to the end or it is still in the middle, right? So that's method we use. So here, ensure, the answer is ensure consistent result. It is important to use a method other than reading from eyes, such as color matching. This helps to standardize the final color, as color perception can be subjective. Because question is asked not to propose a method, explain the method. That means you had to tell the method, okay, color chart. The reason is, explanation is, this is subjective, so this gives accurate result. Understood? Okay, next one. These are the market scheme. I just show the main market scheme point. Use a color matching chart because it's a subjective. Other information student can always propose. I removed. We will do it later. Question number 2. John wanted to investigate the effect of temperature on the rate of carbon dioxide production by yeast. He set up this apparatus. So you can see in this apparatus, there is a water bath, and inside the water bath they place the student placed the test tube, and inside the test tube you have a glucose and yeast, and the top layer is covered with the oil, and it is connected to the another test tube, and we can see bubbles coming when the reaction is active. So that is the instrument can use the yeast activity, because yeast respire, and when yeast respire they produce carbon dioxide. So carbon dioxide goes out of the tube as bubbles. So then we can count the bubbles, and we can also measure the temperature of the water bath, and we can record how many bubbles come with this much temperatures. Okay, the oil layer prevents the entry of air into the glucose solution. Explain why this is necessary. So you can write the answer and pause the video and write the answer and check with me. So oil layer prevent the entry of air into the glucose solution. Explain why this is necessary. Okay, go ahead and write answer. If not, I will continue to show the answer. Now, when the test tube is covered with the oil layer like this, right now this is yeast and that's a glucose, right? So now yeast take up the glucose and they do respiration, and this respiration is happen in this solution, and if oxygen wants to come to this place, it cannot, because of the oil layer prevent entry of the oxygen. So here the oil layer prevent entry of oxygen, right? So that's a main point. Oil layer prevent entry of oxygen, and then yeast can respire anaerobically. Yeast can respire anaerobically, right? So that's a answer for this question, and you can see a model answer. This is important to prevent entry of oxygen, then it maintain anaerobic conditions. Market scheme point: keep out oxygen, anaerobic, prevent aerobic So anything come under these points, then you get the marks. Then next one, John varied the temperature of the water bath between 15 and 65. Now he changed the temperature 15 to 65. He measured the rate of carbon dioxide production by counting the number of bubbles per minute. Sketch the shape of the graph that John would obtain on the axis below. So that also we learned previously. So if we have different temperatures, what will happen? Enzymes are affected. So 15 to 65. And that's a rate. So all the reactions are catalyzed by enzymes. So we change the pH, enzymes are affected. So we know that optimum pH, and then after that, it's like this. So the graph should be like this, because temperatures, high temperature denature enzyme and low temperature kinetic energy reduced and less rate of reaction. As a result, anaerobic respiration is also affected. So this is the answer. You can draw something like this, it's fine. And give the dependent variable in this experiment. We learn a COMB scheme. Under the COMB scheme, now you can play what is a dependent variable, what is independent variable, what is control variable. So dependent variable is what we measure. So now you can write the answer, what we measure in this experiment. So you can pause the video, now you write the answer. Okay, so now I am continuing the video. So what we measure in this experiment, so here you can see the points. So you can see here the change. So what we change is the independent variable. So that means when we are changing something in the experiment, that is independent variable. Now organism, if we have a living things, we have to also concern certain points. They should be same number, same size, like that. So organism is yeast. Then any experiment we have to repeat, and we have to take measurements. For example, in this case, we can measure two things. One is number of carbon dioxide bubble produced, measurement number 2 over 5 minutes, right? So time and the number of bubbles. And the standardizing other variable or control variable, we should keep other variable same, right? That's a very important thing to remember. Try to understand this, try to remember this at least every week until you get this scheme. You have to work on it, right? So number of carbon dioxide bubble produced is the dependent variable what we measure. Give the independent variable in this experiment. Now you can do it, pause the video and write the answer, right? Go ahead. I will continue the video. So independent variable is what we change. We change the temperature. So one hint I will give to guess independent and dependent variable, always in a graph, x-axis is independent variable, y-axis is dependent variable. In a graph, x-axis is always independent variable, y-axis is always dependent variable. So suggest one way that John could improve the reliability of this experiment. Now reliability is the, in experiment, how reliable the result. You measure one time, maybe something went wrong, your results are wrong. So you measure second time, so then you get the values. Then you measure third time, fourth time, fifth time, sixth time. So if you've done 6 times and your result are very close to each other, but one may be something wrong completely, or it's too high or too low data. You remove this. This is called anomalous value. And if the values are same, you get the mean value. That's a reliability is repeatability. So repeat the experiment, right? Last thing and few things suggest how John could improve the accuracy of this measurement of the rate of carbon dioxide production. Now John measured number of carbon dioxide bubbles. So he missed maybe some bubbles. Some bubbles are smaller, some bubbles are bigger. So it's not accurate. So now if we want to measure exact volume of carbon dioxide in this kind of experiment, only way is, or not the best way, we use syringe. Fix a syringe and measure the volume of the carbon dioxide. You can see here the syringe is fixed to the end and we can measure the carbon dioxide volume. Last thing, give two variables that John would need to keep the same in his experiment. Now, temperature is independent variable and dependent variable is carbon dioxide. Now, other variables you can propose like this. Example, the glucose, volume of glucose solution, concentration of glucose solution, number of yeast cells, mass of yeast cells. So these are the things we can propose, and here Martin's scheme proposed mass, volume, number, yeast species, type, yeast, pH of solution, right? So next one, write the word equation for anaerobic respiration, and I think all of you can do this. Go ahead and check the answer with my answer, whether it's correct or not, right? So here I'm going to show you the answer. So anaerobic respiration in yeast, you can see here glucose, and it converts into alcohol and plus carbon dioxide. Okay, so we have done our first HOG homework with the online guide. So you complete this work and hand over to me in the next class. I go through everyone, whether they have done it correctly or not, and I will give a feedback on the homework sheet and return to you in the next class. So thank you very much.